Good afternoon, everyone. My name is Sloan Sink, and I am going to be speaking about my research done in southern Vancouver Island on English holly to determine whether or not it changes soil biology and chemistry. Before I get into that, I do want to give a land acknowledgements to the land that I was working in and where I am presenting from today. So I did my research in the unceded territories of the Lekwungen speaking peoples, which is in southern Vancouver Island, and includes a wide variety of different groups of people. Mainly, I do want to acknowledge the Shawnee First Nations, where the research was mainly conducted on, which is in Metchosin, the Metchosin Park, as well as the Matheson Lake Regional Park. When doing ecology, one thing that I learned in school that I think should be expressed throughout all science, but especially ecology, is the importance of acknowledging the original people that lived in an area and the effects that colonialism has on the native ecosystem, because it is our job to try to bring these places back to either their original health or a reflection of that health so that we can cultivate the plants that used to live in these areas and also just show respect to uh, traditional values that have been in time immemorial have been developed to work with and sustain these very special places. As well, I am presenting from Oahu on the island, on the Hawaiian islands, and I also will acknowledge the Kanaka Maori, which are the original Hawaiian people of the land. And I do hope that this research can be applied to invasives throughout the world. Thinking about invasives on where I am, I started working on my own property with an invasive called Halicoa, which is it is a similar kind of invasive to English holly, which I will be talking about soon, is in that it is a woody invasive and it grows very quickly. So a lot of the practices, or the main practice to prevent growing that is used by the Machosen Invasive Species Cooperative, who provided me this research opportunity. That method of killing and suppressing invasive Halicoa that we have here in the Hawaiian Islands is also applicable. So I think that it is nice to find ways that we can incorporate the knowledge that we take for invasive species that are difficult to deal with and find ways that we can potentially use them in other invasive species. There are many people who have helped me with my project and who have provided encouragement to me, and I appreciate all of you. I do want to point out first Professor Nancy Shackelford, who is my project advisor from the University of Victoria and is the associate professor and director of the Restorations of Natural Systems School at UVic. She was able to connect me with the people that I needed and provide guidance, but enough independence that I felt like I really did a lot on my own, as well as Brooke Hayes, who is a PhD candidate also at UVic in the gastronomy department, providing lab equipment that I use for my research, as well as giving me just so much knowledge that I could incorporate in my research and make sure I knew how to use the equipment I was provided. Thank you to the Chosen Invasive Species Cooperative MISC coordinators Neil West and Brenda West for guiding me through connecting with the parks I did my research in and giving me background information on English holly, as I have had MISC for a few years since 2019, and this has been their main goal in research. Thank you to Admin Officer Bob Payette for helping me get Indigenous approval and understanding of what I was doing in the parks before I went and did that. as well as Professor Stephen Businger from the University of Hawaii in the meteorology department for looking over my paper, giving me advice on how to change it. Thank you to Deputy Corporate Officer Tina Hansen from Machosen for getting me the permit I needed to work in the parks, to forest ecologist and botanist Andy McKinnon, who was very enthusiastic about my work. And when I did my presentation in person in Machosen, came and did an introduction speech for me that was very nice. Thank you to my friends and family. There were a lot of friends and colleagues who came out and assisted me in field work, and that was very helpful because I worked on this project for 1.5 years, and field work took in total, probably 2 weeks. Well, actually, no longer. It was very hard, but very fulfilling, and it was nice to have friends there with me to help. I did get some funding support from the Habitat Conservation Trust Foundation and the Machosen Invasive Species Cooperative together, as well as the University of Victoria, the Laureen Kennedy Grant. So the title of my research and my paper that I'm working on is the impacts of invasive English holly on soil chemistry and biodiversity through the restoration of natural systems, and this lasted from spring 2024 to 2025. I'm just going to move my photo here to make sure we can see all of my notes. So Vancouver Island, Southern Vancouver Island is a part of the coastal Douglas fir biogeoclimatic zone where we have 100 unique plant species from shrubs, trees, mosses, herbs, and vines. And when we are looking at these unique biogeoclimatic zones, it's important to remember that this exists here, and the things that evolve here have evolved to live in this climate, and they're unique, and they deserve to continue to live in these places because it affects everything else around them. And since it is unique to us, why wouldn't we want to keep it around? And that kind of motivation is what propels me through restoration. So my project was motivated by when I actually went to the parks and I was able to see the creation of dead zones, which are underneath English holly trees. The living tree, you can see where the extent of the canopy is, and at the bottom there's kind of a ring of just bare topsoil where nothing is growing or very little plants are growing. And even in sites where MISC took me where they had cut the tree, treated it, and it had been treated for 2 or 3 years, there were still these rings of dead zones. So clearly there had been long-lasting impacts on the soil. And to see something that clearly there's something wrong, it is very emotionally and scientifically rewarding because I can imagine there is something happening here and there's a reason to try to do the work, and the goal is to make it better. So ecologists, members of community, and government officials should care about our ecosystem. So my hypothesis is that invasive English holly alter soil chemistry and physical properties underneath tree canopy drip lines, affecting native ecosystem health and integrity. English holly is a perennial tree, and it can grow up to 15 years of maturity. It is notable by the leaf forms that are quite thick and waxy leaves that are a little spiny. They typically have red berries, as shown in the image on the right here, which is on someone's property, but they can be yellow and orange as well, and they come in bunches. The flowers, which are present on male and female trees, are white. And so they are dioecious species, as in that image there. That is a female berry producing tree, which is huge. They can grow so big, and they are very popular for as a decorative, beautiful ornamental plant, which is where their introduction to Vancouver Island comes from. English holly has proliferated throughout the West Coast, such as British Columbia, California, Oregon, and Washington, as well as dispersed in other sites of the continent. But I just wanted to stick here because this is most important to my research. And the Invasive Species Council of Metro Vancouver identified English holly as one of out of 12 of the most problematic invasive species, notable for its challenges of removing, especially once it gets to this huge point. And English holly does look similar to Oregon grape, Mahonia nervosa, which is native. So I just wanted to show a comparison. When English holly is small, like 1 or 2 meters, it may look like the image on the left here. The main difference is that the berries are red versus blue, which you see in Oregon grape, but also the leaf forms, they're kind of dispersed asymmetrically versus that symmetrical um, mutation we have on the right there. The leaves of Mahoney Universe are quite thin comparatively. It's not as spiny if you were to fall on it, which I have done in the field. So I know the difference in how it feels to fall on these plants. So my initial observations that support the hypothesis is dead zones, of course. And through research, I found a lot of research talking about invasive species that change soil chemistry to increase their success through root and leaf exudation, as well as supported by the novel weapons hypothesis, which simply states that invasive species that are very successful change soil chemistry or change physical properties of the environment to suit their benefits, which makes sense, or suit their needs. Why should we be concerned about English holly versus any other invasive species? What makes it of equal or more concern? As mentioned, dead zones. These are certainly the most important part to consider, which is the visible patches of bare soil underneath holly drip lines. So that's under living trees, it's under treated sites. Research by Stokes et al. In 2014 found that 79% of area under holly canopy is devoid of native evergreen and woody vegetation, which does also reflect in my research. And, invasive species that cause soil changes in soil chemistry are, as at least pointed out by several researchers, and their opinion is of highest concern. And I would agree with that just because of the long-term implications it has on the ecosystem. And this image here, taken by Brenda West from MISC, is an example of a treated site that is 2 years old, but still has the existence of this dead zone here. So another concern is that it is a woody invasive species, and woody invasives have a specific growth pattern. So there it starts with a lag period where an invasive is introduced to a site and it's growing relatively slowly. It doesn't seem of very high concern. It's not forming huge thickets of plants that grow really densely in an area. And this slow growth and spread can last for 10 or more years. But then it goes into this exponential growth phase shown on that graph there, where there is a rapid sudden growth of these species. Model done by Stokes et al. On English holly showed 1 year that the plant cover was 0.2%. After 10 years, it reached 2%, 25 years to 15%, and 35 years to up to 50% or more. So you can imagine if an area, if one of these parks it went through the woody invasive growth rapid exponential growth phase. After 35 years, it could be 50% or more inundated by English holly. English holly also has a close to zero mortality rate after it reaches 10 or more years, because that it takes about 15 years to reach full maturity, but once it gets to 10 years, it is typically quite big. Um, And if you couple that with the potential that it's changing soil chemistry and it's taking over huge swaths of land, that is concerning. So, due to the initial slow growth and lag phase, invasive English holly may go unnoticed until the exponential growth phase and population boom. It disperses through suckering, which is allows the plant to actually grow from roots that come out of the main trunk of the tree. It can be cut and branch off into more. And English holly also forms a taproot. And when you try to take out the tree, if you break off the taproot, it will just sucker and create more underneath. It also is dispersed by birds, especially American robins, European starlings, blackbirds, and chickadees. And these berries are produced year round, so it is a constant source of food. Although the seeds are only produced on dioecious female plants, and it is a fair point from birders that if we were to remove English holly, what would be available for birds to eat? But there are a lot of native plants that provide food as well. And some of those include arbutus, blue elderberry, cascara, coastal strawberry, Pacific dogwood, red flowering currant, red osier dogwood, salmonberry, and thimbleberry that are shown to be commonplace for American robins to stop and eat. And so that provides a bird-friendly relationship with the native ecosystem, which is something we want to cultivate rather than proliferating this invasive species when we can easily replace it by something that is native. So how did I conduct my research and what did I do? So I went to two parks, the Machosen Wilderness Park, where here I have waypoints in white, which represent control sites. It's randomly chosen that represent what should be expected throughout this park. And then treated sites are in pink. So there are five treated and 10 controls. Treated are sites that are 2 or 3 years treated, where the English holly tree has been cut and treated with garland at the stump, so it doesn't continue to grow. I have 10 more control sites at Matheson Lake Regional Park and five living tree sites. They're all in the same area, but those were very large trees, so they're very good examples of English holly soil change. And this just shows you vaguely where they were, relatively close to each other, so the soil conditions should be relatively similar. Methods and materials. What were the test methods I conducted? So I did the tea bag index where I would take red and green tea and place them in the soil to measure bacterial activity and fungal activity. The tea bags are made of a synthetic tea bag so that it doesn't decompose, but only allows microbial activity to enter the tea bag without tearing it apart, so I can measure the weight afterwards. So green tea bags will measure decomposition by microorganisms because it is more readily decomposed as it is a labile material. It's softer and it will show stabilization or S. A rooibos or red tea is more recalcitrant and it's a woodier tea, so there's a higher weight that will be left over, but it shows a decomposition rate measured by K. Total organic carbon was also measured through soil that I took from the site. I would incinerate it and then weigh before and after to get a sense of the amount of carbon that was left in the soil, which shows retention of carbon and soil health. Typically, there's about 58% of the organic matter is going to be made up of carbon, so you can see the range. And plant surveys, which is a semi-quantitative measure that would show me the amount of plant and in a site, as well as the percent cover of a site. So you may have a site that has one species, but it's like 95% covering a site, like Salal, or you could have four species and they each cover only like 10%. So there's an area of 60% that's just bare topsoil, for example. And so I would take a cross section here. Each square is 1 by 1 meters, and I would look within each of these squares to determine, okay, I have some um, sword fern here, I have some sword fern and salal here, I have sword fern and salal, and then I'd go up, and that would be the measurement. Now, my results and interpretations next. So my tea bags index, I found that tea by weight in grams was highest under alive sites about and control and treated were about similar here. Green tea was less at controls and less at red for red tea as well with controls, but it was relatively close to treated. So what this likely means is after 110 days of incubation in the soil, starting with the same weight to begin, we see a reduction, and yet the alive sites, living holly sites, had a higher amount of tea that was left over than the controls. So there is less consumption of the tea. So perhaps this means that um, the type of organic matter that is introduced into the soil is harder to decompose for microbes, so there is less amount of them that are actively trying to decompose. It could also be that the amount of microbes is reduced because they are having a harder time to actually decompose this tea and it's taking longer. Um, And that would likely be due to root exudation. So the exudation of some kind of chemical that is in benefit for an English holly tree. Total organic carbon was least at alive sites and actually highest at treated sites. And total organic carbon will represent higher nutrient retention, cation exchange capacity, cycling and microbial activity, and higher water retention as well. So the lowest total organic carbon percent at a live sites mean that it is struggling with those in comparison to total or sorry controls and to treated sites. And I think that it's possible that treated sites have the highest amount of total organic carbon because there's kind of like a rebound effect happening where the tree has been cut down and treated, and there is no new organic matter being pumped into the environment because there's little things growing on the surface. And so it's trying to balance out what is left there that needs to be consumed. And so there may be a point where that will actually plateau. It's unclear. It's hard to say at this point just because of my sample size is relatively small, but that is a potential. And species richness and cover, we saw that there was less plant species overall at alive sites and lower plant cover by percent at alive sites as well. There is, I will note, a variability within the control site of the amount of coverage. And this is something that I would need to have a larger sample size to be certain about these numbers, but there is a relationship that we can see through the means. So poor soil health for native and non-native understory species is going to lead to less likelihood of a plant being able to survive in this area. So in conclusion, let's go over the results. So for one, T weight is highest under alive and lowest under controls. Decomposition K is slightly lower under alive and the same under treated and controls. Stabilization S is a measure of long-term carbon storage is lowest at alive and highest under controls. pH is highest under treated and lowest under alive. TOC is lowest under alive and highest at treated. Species richness is lowest under alive and highest under treated. And percent plant cover is lowest under alive and highest under controls. So the significant findings here, low K and high S at alive. So that means that the organic matter that is available there is resistant to decomposition, and when it is decomposed, it is simply being stored and it is not being consumed. So we also notice that there's reduced plant cover and species variety at the living sites in particular. There's low total organic carbon or soil organic carbon at living sites. And likely our results show that there is a reduced microbial activity because of the higher amount of tea that is left over. So these combinations result in slow initial decomposition of material and reduced organic material stabilization, which could lead to actually potentially carbon that is being re-released into the atmosphere more easily than it is being absorbed in the soil. So what could cause this? I think it's root and leaf exudation, which is supported by the novel weapons hypothesis that just says that a species, especially an invasive species introduced to a site, wants to control it and change it to its benefit. And that just makes sense. So invasive organic compounds can change substrates. So these are allelopathic defensive antimicrobial chemicals. A member of the Ilex family called Ilex asperella has been shown to exudate triterpenoid saponins from its roots. And those same triterpenoid saponins are found in English holly berries. So there could be a relationship there. As well, when you combine the exponential growth pattern of an invasive English holly as a woody invasive species, you have large areas of land being experiencing soil chemical modifications and just total out competition of for native species because it is completely wiping these areas of any understories plants that you can see on the right there. So, what is being done to prevent or spread information about English holly? So we have English Holly Management, and this is being mainly spearheaded by the Machosen Invasive Species Cooperative, or MESC. So they have found a very um, conservative way to use pesticides called Garland, where they actually covered an area of 230 plus acres of parkland, which is supported by the Habitat Conservation Trust Foundation, where they would cut the stump cut and treat the stumps with the very clever idea of using an actual dabber, so you aren't spraying the site and using excessive pesticides. You use it directly on the site, um, and that ends up saving you money, preventing these sorry herbicides. I mean these herbicides from going excessively in the soil, and puts it directly where it needs to be. Of course, if you have a sapling or a relatively small one, you can pull it up with a polar bear. As long as you actually get all the roots out and don't leave the taproot in, it will not come back. And provincial government awareness. This is slowly we have been seeing changes in the government. Chosen very much driven by MISC to actually open our eyes and start making changes in order to remove and eradicate or at least get a handle on English holly. And one thing that Neil West from MISC said to me, which I think is very true, is that English holly takes 15 years to mature. Therefore, 15 years, we have 15 years to get it under control. So this is one of those where, like he said, in reflecting with other invasive species, it's hard to imagine getting control over it. But English holly, we do have a chance because it is something that we can go in and we can make a difference as long as we have the support from the local government to allow us to make the changes we need to. It would be it's totally possible. And I think that it's important to start doing this now before we get into that exponential growth phase and English Holly becomes unmanageable. Future research. I think the most important thing would actually be to look at the microbial impacts and whether or not the microbial community is suffering from English holly, because that is a very essential, just first step into understanding the soil chemistry and biodiversity of the area. As well, I would like to see a larger sample size be done so we can have more accurate research and but we need more focus on this area in order for us to get that opportunity. Thank you very much for listening. And if you have any questions, you feel free to email me. My email is sinks@hawaii.edu. I will be publishing my paper, working with Nancy Shackelford on that. And once that is available, Neil West and Brenda West will have that on their website at MISC. So you're welcome to read about that. And mahalo from Hawaii.